Here we come to finish the unfinished part of the last part of grade 11 biology. Unit 2, that is about osmo regulation. Therefore, I think we stopped here, the formation of urine by kidney. Therefore, the first step in urine formation by filtering blood is the filtration part. The ultra filtration, they call it pressure filtration, because almost about 15 millimeter mercury pressure, net pressure, is created in the Bowman's capsule, in the glomerulus in the Bowman's capsule, that helps to filter substances from the blood, except of course everything comes out except red blood cells, white blood cells, platelets, and plasma proteins, or proteins that are found in the blood. Therefore, this is the first stage. The first stage is ultrafiltration or pressure filtration, also known as glomerular filtration. This filtration process is the first step in urine formation by kidneys, and it occurs in the Bowman's capsule. Here you can see the Bowman's capsule, and you can see one arterial bringing blood into the network of capillaries known as glomerulus in the Bowman's capsule, and then a narrower artery, arteriole, of course, taking blood after it has been filtered by pressure created there inside. Therefore, there are filtering layers of two layers of cells in the under the glomerulus, two layers of cells and basement membrane. These are the filtering units of the kidney here. Therefore, the blood that comes into the glomerulus in each Bowman's capsule comes through afferent arteriole, which branch into capillaries inside the Bowman's capsule. Capillary mass, special name is given to this mass of capillaries, glomerulus, plural glomeruli. And then out, the blood is out from the glomerulus through efferent arteriole, the less diameter arteriole. You see here nature against the network of blood vessels in other parts of, in most of the other parts of the circulatory system, that is, arterioles give rise to capillaries, and then capillaries merge together to produce the smallest veins, venules. Here, this capillary network is found between two arterioles, still arterial systems. This is nature made this purposely in order to maintain the high pressure tubes. Arteries or arterioles are high pressure systems, so that high pressure is needed in the glomerulus in order to filter blood. Plus also, not only being in the arteries, which maintains high blood pressure. Look at the diagram. The diagram, the afferent arteriole has wider lumen, that means wider diameter, whereas the efferent arteriole has narrow. So blood comes through wider tube and tries to come out through narrower tube. This by itself increases the pressure in the glomerulus. Therefore, these two adaptations: one, the fact that glomerulus is being held between two arteries. Of course, arterioles are the smallest arteries. Yeah, arteries, plus arteries being high pressure system tubes, and also the afferent arterioles being wider than the efferent arterioles. These two together create a net pressure of about 15 millimeter mercury, which is responsible to filter blood in this Bowman's capsule. And this is the first stage that we give the name ultrafiltration or pressure filtration or even glomerular filtration. Therefore, then within the blood in the glomerulus in the Bowman's capsule, high pressure is produced because of two reasons. There are reasons here. Two reasons. One, as I've told you already, One, the capillary mass called glomerulus is formed between two arterioles, afferent arteriole and efferent arteriole. You see, artery is high pressure system, so it helps to produce, it helps to maintain high pressure within the capillaries if the capillaries are in between arteries. This is the case. The second case is reason, the second blood coming, the second reason is the blood coming to glomerulus comes through afferent arteriole, which has wider diameter, and leaves the glomerulus through efferent arteriole, which has narrow diameter. Thus, blood coming through wider tube and leaving through narrow tubes results in a build-up of pressure. You see, these two factors or these two adaptations help to build high pressure, net pressure, that pushes the fluid into the Bowman's capsule, the space in the Bowman's capsule. You see, so that the blood is filtered and the blood is forced to lose its contents. We shall come to that point. These two together create a net pressure that forces the contents of the blood, except the blood cells, platelets, and plasma proteins, out of the blood into the space of Bowman's capsule. You see, in this first stage, the blood is forced to lose all its content, its water, electrolyte content, its metabolic wastes, except of course from the blood components which can't pass by this pressure filtration. The Bowman's capsule are, as far as the kidneys are normal, these elements of the blood do not pass out to become filtrate or glomerular filtrate. One, blood cells, both red blood cell and white blood cells, platelets, and plasma protein. See, therefore, the pressure pushes all the others except this one into the space of Bowman's capsule. The process by which the fluid is filtered out of the blood by the glomerulus is called ultrafiltration or pressure filtration, as we have mentioned earlier. And the filtrate made is known as now the filtrate. The filtrate, the fluid that has been forced to come out of the blood. The filtrate is known as GF or glomerular filtrate, and it mainly contains one glucose, amino acids, salts, water, metabolic wastes, and all these, except of course, it contains all contents of the blood except red blood cells, white blood cells, platelets, and plasma proteins like albumin, fibrinogen. These are very big molecules. They are not allowed to come out of the blood into the space in the Bowman's capsule, unless, of course, in case your kidneys are abnormal, failing, proteins can come out, proteins, so that the proteins may appear in urine. Even red blood cells may appear in urine, which is known as hematuria. Hematuria means urination of blood, protein urea, urination of protein. Of course, in case of failure of kidneys, all these leukuria, even leukuria, leukocytes are white blood cells, so that if white blood cells are found in your urine, there is a problem in the filtering mechanism of your kidney, which could be indicator of failure of kidney, of course. This is again Bowman's capsule. You can see to the right the blood, the blood in the Bowman's, I mean the blood in the glomerulus. Then under the glomerulus blood vessel, there are layers of cells, two layers of cells. One group of cells are very important here. The blue ones are known as podocytes. Really, they are smart. There is also basement membrane. All these layers are responsible to filter blood due to pressure, so that you can see when the filtrate comes down, and it filters into the space, the Bowman's capsule space. The blood entering the glomerulus is separated from the space inside the Bowman's capsule by two cell layers and a basement membrane. These two cell layers and basement membrane are the shifts to filter the blood, and only the small soluble molecules can come through due to the high pressure in the glomerulus. Blood cells, blood proteins, and platelets and enzymes are also proteins that are found in the blood, are not allowed to pass and become GF or glomerular filtrate. The second process, now we are done. Now we have filtered the blood. The blood is losing, of course. If there is glucose in the blood, glucose comes out. Amino acids, nutrients are also lost. Water, which may be needed in the body, electrolytes or salts. The glomerular filtrate contains useful substances such as amino acids and glucose, and also most of the salts and the water filtered out of the blood at the Bowman's capsule in the first stage of urine formation may still be needed in the blood. You see, that's the second stage works. The second stage, which is known as selective reabsorption, the second stage works to return the contents of the glomerular filtrates that are forced out of the blood but needed in the blood. And this second stage of urine formation is known as selective reabsorption. You see, as the GF, called the GF glomerular filtrate, move slowly through the tube system, the different parts of tube systems. You see, tubular part. Nephron has Bowman's capsule, then tubular part, and the tubular part, the tube systems, convoluted tube systems, proximal convoluted tube system, U-shaped tube section, which is known as loop of Henle, and the distal convoluted tube, and finally there is a collecting duct to which the distal convoluted tubules of many nephrons are attached. Collecting duct is not for one neuron. There are millions of neurons which are attached to one collective duct. Therefore, the flow of the GF is from Bowman's capsule space to proximal convoluted tubule, then to loop of Henle, then distal convoluted tubule, then to collecting duct. From collecting duct, it drips into the pelvis part of the kidney. From the pelvis into ureter. From ureter, it goes to the bladder for temporary storage until it is peed or urinated out. You see, therefore, the second stage is selective reabsorption. This selective reabsorption is done as the filtrate, which filtrate? The GF, glomerular filtrate, passes through the tubular part of the nephron, as I have told you earlier, the sequence of the passage. Generally, during selective reabsorption, all the glucose and all the amino acids are absorbed. Yes, definitely, unless the person is diabetic, and had a large amount of glucose in its blood, so that the kidneys are unable to reabsorb beyond their capacity, so that some of the glucose may pass out through urine. And the second, most of the water is also returned back to the blood. Most of the salts are absorbed as glomerular filtrate moves within the renal tubules. In the process of selective reabsorption, different parts of these tubules have different different roles here. Let us see briefly just what each part of the tubules do. Now the first, the longest, most of the absorption is done here. Proximal convoluted tubule. They call it proximal because it is the nearest to the Bowman's capsule where filtration occurs. The first part of the renal tubule, and also it is the longest part of the tubule where most of the reabsorption process occurs. As the GF, glomerular filtrate, passes through the proximal convoluted tubule, all the glucose, look, if you are healthy, all the glucose, all the amino acids, about 60 to 65% of sodium chloride, about 65% of water are reabsorbed by the cells surrounding the PCT, proximal. By the way, this The cells that surround this tube, the proximal convoluted tube, to have um, villi, just like the villi in the digestive system to increase surface area. Therefore, these cells also have adaptation to increase surface area. For what purpose? To undergo efficient reabsorption from reabsorption of water, salts, glucose, amino acids from the GF that passes through them and enter the reabsorbed and enter into the blood in the peritubular capillaries that surround the tubules. Yes, efferent arteriole again produces capillaries, and these capillaries produce a bed network of capillaries around the tubules, with all the tubules, the bovenal, proximal convoluted tubule, distal convoluted tubule, and even collecting that. Therefore, in order to accept what is going to be selectively reabsorbed and in order to accept or take what they have forcefully lost in the Bowman's capsule because of the pressure, they again surround the tube systems, and this network of tubes, network of capillaries that branch from efferent arterioles are known as peritubular capillaries. Therefore, selective reabsorption, the cells of PCT selectively absorb and return the reabsorbed material into the blood in the peritubular capillaries. The second part is lupophenol. The lupophenol is a U-shaped tube that extends down into the medulla of the kidney and turns up. Of course, it has two parts, therefore. The part that moves down materials is known as descending limb. The part that moves the glomerular filtrate up is known as ascending limb. Descending limb is highly permeable for water. Therefore, water, it reabsorbs water again. You see, the remaining, if the body needs the body needs to reabsorb water, therefore, the descending part of the limb of Henle is specially adapted for freely permeable for water. Water is reabsorbed additionally here. As you can see, therefore, the filtrate, the GF, as it is moving through the descending limb and losing water, it becomes more and more concentrated. The ascending limb is more permeable to salts and less permeable to water. Salts, especially chloride salt, sodium, calcium, magnesium, if they're therefore the ascending limb is the area where reabsorption of these salts is made. You can see this diagram. This is one nephron. You can see the U-shaped part, ascending and descending limb. Thus, it has descending limb and ascending limb. Descending limb highly permeable for water and is responsible for water absorption. The ascending limb is more permeable to salts and not less permeable to water. Of course, it's a repetition, redundancy. Yes, of course. The third part is distal convoluted tubule. We call it distal because it's very far from the Bowman's capsule. The cells, convoluted means twisted, of course. The cells around the first part of the distal convoluted tubule are influenced by aldosterone. Remember, this aldosterone is a hormone produced by adrenal cortex, the outer part of adrenal gland, aldosterone. Aldosterone is produced when your blood sodium level is low. Blood sodium level is low. That means when there is hyponatremia of blood, the blood sodium content is low. So this stimulates adrenal cortex to produce aldosterone. Aldosterone through blood comes to the distal convoluted tubule cells, the first part of distal convoluted tubule cells. Then they stimulate these cells. Please. The blood of this personality or this man or woman is low in sodium. Don't allow sodium to pass into urine. Therefore, this part, the first part of the cells in the first part of distal converted tubule reabsorb efficiently reabsorb sodium and return it into the blood. And as sodium is being reabsorbed, water follows by osmosis. Of course, water is also water follows the salt. They say, yeah. At its end, you see, we say the first part is influenced by aldosterone to reabsorb what? Sodium. At its end, the last part, the tip, the distal converted tubule has specialized cells known as principal cells. These cells are stimulated by another hormone from the posterior lobe of pituitary gland, and this hormone is known as ADH, antidiuretic hormone. Don't forget ADH, antidiuretic hormone. And this hormone stimulates the principal cells so that principal cells are efficient and reabsorb much more water. Therefore, ADH is produced when our blood is dehydrated, when still the blood needs higher amount of water. The blood your body might have lost or may be losing large amount of water by sweating because of the external temperature. So you should not also additionally lose large amount of water in your urine. ADH is produced in case of dehydration of your blood, in case of hypertonic blood, low water but much solute, highly concentrated blood. Uh, ADH is produced and released into blood, comes to the kidneys. Which part of kidney? Of course, not only distal converted tubule. We shall come to that point. Collecting duct is also stimulated. Cells in the collecting duct also contains principal cells. These principal cells are the target cells for ADH. ADH acts on the cells, and so that the ADH makes these principal cells to reabsorb maximum amount of water from urine, so that during ADH production you produce concentrated urine because you are returning much water, not to be urine, of course, conservation of water. This is another diagram as you observe, you see. The Bowman's capsule proximal converted tubule is found in the cortex part of your kidney, the outer portion. Then the other lupophenol, most of the collecting duct, of course, proximal converted tubule, small part of the loop of the first part of the very small portion of the first part of loop of Henle and distal converted tubule are all found in the cortex of the kidney. The collecting duct maximum part is in the medulla of the inner part. We may divide it outer medulla, inner medulla. You see this diagram. Therefore, you can see what has been filtered here in proximal convoluted tubule. You can see that water reabsorbed sodium, even ammonia is also secreted. Ammonia can be secreted. Ammonia is secreted. That means from blood surrounding the tubules, if there is the blood contains ammonia, that is toxic and given to the tubes. There is the third stage of urine formation, which is known as secretion. What you observe here, substances given to the tubules from the outside environment, this process is known as secretion. Selective reabsorption finish when substances move to blood from the glomerular filtrate in the tube systems, you see. The fourth collecting duct. The collecting duct contains the principal cells that are stimulated by ADH. For more water reabsorption, the same is true. We have said this, of course. Therefore, they are very important. Collecting duct and the last part of distal converted tubule are very much responsible for. They are targets for ADH, antidiuretic hormone. By the way, this hormone is also known as vasopressin because of the fact that it has another role of pressing, narrowing blood vessels, which is known as vasopressin. It narrows blood vessels in order to increase pressure in case of decrease pressure. Its main role is in water conservation by acting on the principal cells, and these principal cells are found in the two tubular parts, the proximal last parts of the distal converted tubule, and the collecting ducts contain principal cells. Therefore, principal cells in the kidney are the targets for ADH. When the body is dehydrated due to excess loss of water, the blood becomes hypertonic, and hypothalamus checks blood tonicity or blood concentration as it passes through it. And if it is hypertonic, that means hypertonic blood means it has very minimal amount of water and much solute, concentrated blood. Hypothalamus stimulates the posterior pituitary to release ADH or vasopressin, which then acts on the principal cells in the last part of the distal converted tubule and in the collecting duct, causing more water reabsorption so that only very small water is lost as urine. Water conservation. So you produce what? Concentrated or hypertonic urine. Even its color is very dark, uh, darker color because of low amount of water. Thus, a more concentrated hypertonic urine is produced. This is the essence. The third stage, tubular secretion, third process of urine formation by which substances move from blood in the peritubular capillaries into the renal tubules. Of course, this is minor stage, minor event in urine formation, but still, if there are substances to be uh, given to the tubular systems of the kidney, so that they will become urine, part of urine, still the network of blood vessels surrounding the tubes, known as peritubular capillaries, secrete substances into what? Into the renal tubes, either proximal or lupophenol or distal or collecting duct. You see, all these tubes are surrounded by capillary network. You see, this is known as tubular secretion. The blood is secreting substance into the tubes. For instance, in the distal converted tube, there is secretion of H plus in pH regulation. Now, for example, look, H plus is coming from where to where? H plus is secreted means it is taken out of the capillaries, the blood in the capillaries, and given to what? The distal converted tube. This is in order to adjust, regulate um, pH, pH, pH of blood. If the pH of blood goes down, it is because of higher amount of H plus in the blood. So in order to bring it to normal, H plus is secreted from blood into the tube so that they appear in your urine because they are excess. Therefore, we have told that one other role of kidney besides serving as excretion osmoregulation, it also adjusts blood pH, and also secretion of K plus in salt regulation. Salt regulation when regulate when there is excess potassium in the blood. Peritubular capillaries also give potassium to the tubes. Note, urine is formed constantly in the kidneys and is uh, it drips to collecting ducts, then to renal pelvis through ureter in the goes into the urinary bladder, muscular sac which can hold between 600 milliliter to 800 milliliter urine. It empties the urine when it holds only 150 to 350 milliliter. The blood, temporary storage. Elimination. The urine can't escape from the bladder because it has two sphincters, rings of muscles, upper sphincter, lower sphincter. The upper sphincter is involuntary, whereas the lower sphincter is voluntary. When the sphincter muscles relax, the muscular walls of the bladder uh, expel the urine through the urethra, and the process known as peeing or urination. Adults can control this sphincter muscle and relax it only when they want to urinate. In babies, the sphincter relaxes by a reflex action set off by pressure in the bladder. By 3 years old, by 3 years old, most children learn to control the lower sphincter voluntarily. Always the upper sphincter is, of course, involuntary. Whenever there is pressure from the fluid, the urine in the bladder, it opens, but the lower is tight. You can control it, so you don't urinate on yourself. Thanks for lower sphincter. This in terms of composition of blood plasma and urine. You see, blood plasma, water 93%, urea 0.03%, uric acid 0.003%, ammonia 0.0001%, sodium 0.3%, potassium 0.02%, chloride 0.37%, phosphate 0.003%. When it comes to urine, water is, of course, 95% water. The urine is 95% water. Urea, um, about 2%. You see, a huge amount of urea is reabsorbed from the blood. Uric acid is also waste. It is secret. Ammonia is also waste, and sodium, excess sodium can be reabsorbed. Excess sodium can be lost in the urine, of course, because you know that sodium is our daily, sodium and chloride are our daily intakes, salt, especially when you put too much salt, you are going to urinate it. You see, potassium 0.02, plasma 0.15, usually chloride 0.37, 0.6, huge the amount lost in urine. Phosphate is also there, 0.12 in urine, 0.003. Therefore, this is just compare the plasma. Therefore, there are some minerals that are lost each time we form urine. Urine, pH of urine is 4.5 to 8.2, yellowish due to pigment. It contains pigment known as urochrome, which gives a yellowish color to your urine. Smells faint ammonia smell. Volume minimum 500 milliliter, maximum 3000 milliliter. On average, 1000 to 180 800 milliliter per day. This is the production, the amount. A normal personality must have 500 milliliter minimum and maximum 3000. If it is below 500 milliliter per day, it is known as oligouria, low amount than required, low amount than normally required urine you are producing. This may be a sign of many problems, including not drinking sufficient water, or the fact that your food is also not containing enough amount of uh, enough amount of water, or it could be health problem, of course. Therefore, this is the last part of the Osmo regulation. Please stay awake, work hard, and there will be plenty to eat. Please, I will provide you some summary questions on this topic next time, maybe tomorrow, and you work on them and come for your mid exam. Ciao.